Hello, everyone, and welcome to our session about understanding spinal neuromodulation for spinal cord injury. Where are we today and where is it going? So thank you for joining us. This session is hosted by the North American Spinal Cord Injury Consortium. And we just wanted to do a quick introduction to our organization, and then we'll get on with the agenda. The North American Spinal Cord Injury Consortium, our mission is to bring about unified achievements in research, care, cure, and policy by supporting collaborative efforts across the spinal cord injury community. And we do this by identifying the gaps, communicating resources, and really being a conduit for the people with, for the community living with spinal cord injury and the many stakeholders across that throughout North America. We started back in 2018. We have just about 300 members now. We have a very far reach because a lot of the organizations that belong to the consortium are nonprofits that represent people living with spinal cord injury and also individuals as well as other stakeholders. So we believe our network is around 700,000 throughout North America, where we can have a good reach to really allow for collaboration amongst the community. Our organization focuses and operates on projects, and those projects are a lot in terms of collaborating, not just within our community, but throughout the community with our stakeholders, and really truly speaking to our mission about connecting the stakeholders in our community. This session, we're actually going to be highlighting one of our projects, which is a project with the Praxis Institute and our spinal neuromodulation survey to the community. And that leads us into the agenda. So our agenda for this recording is going to feature Peter Grahn from Mayo Clinic and Vivian Mushahwar from the University of Alberta, Edmonton. They're going to talk a little bit about the most recent research. Then we'll bring on John Chernesky from the Praxis Institute, who will talk about this ongoing project that I mentioned earlier about the survey to the community about spinal modulation. We also have a surprise visit from Rodrick from U2FP. He's going to talk just briefly about the quarterly calls that he has with the various researchers and stakeholders within epidural stimulation for spinal cord injury. And then we're going to end that with a Q&A throughout the audience. And this will include one of our other guest speakers, Chet from the University of Washington. So let's go ahead and get on with our agenda, and we're looking forward to a lively discussion. We'll take it away with Peter.
Thank you for the invitation to present here with the group. I'm going to be focused mostly on epidural stimulation, implantable technologies for activating sensory motor networks within the spinal cord below spinal cord injury. Just to kind of give a brief background of the voluntary control aspect that has been highlighted by epidural stimulation in patients that are considered clinically complete, so complete loss of function below their level of injury. Evidence dating back to the 80s shows even in functionally complete injuries, when they did anatomical investigation post-mortem, they found that 84% of these patients still had intact tissue across the injury site. So on the top right here, you can see kind of just a simple schematic by Dmitrievich et al. Showing what a experimentally induced complete injury would look like versus discomplete, where you have some type of spared fibers spanning the injury site, either descending or ascending, versus an incomplete spinal cord injury or partial loss of function. And so epidural stimulation, the idea behind it is that you're basically amplifying the excitability states of the networks that are below the injury site, so that any of these spared connections that are descending may be able to transmit signals that produce or influence those excited networks in order to produce some type of functional output. So this is just a basic overview of both epidural versus, as Chet was talking about, transcutaneous electrical stimulation. So both, when you're targeting the lower extremities for restoration of function, the electrodes are usually placed within the T11 to L1 vertebrae. So this would be a Medtronic 565 array. placed over the spinal cord in this region here that targets the lumbosacral enlargement. So spinal cord segments L2 to S1, which control the lower extremity muscles of the quadriceps, hamstrings, tibialis anterior, and calf muscles. And what's been shown using each of these techniques, so both epidural stimulation as well as transcutaneous, with epidural stimulation, Minazian et al. Showed in supine position when subjects were relaxed and they applied continuous pulses or a non-pattern stimulation, they were able to evoke central pattern generator-like activity, which was these are rhythmic bursts in the lower extremity muscles, as well as cyclic movement of the knee joints. And Minazian et al. With Keith Tansey showed that when you apply transcutaneous stimulation during robotic-driven treadmill stepping, so here this is no stimulation, activation of the lower extremity muscles is very low to none. And when they applied transcutaneous stimulation continuously, you saw the similar cyclic or rhythmic bursting patterns in the lower extremity muscles, similar to epidural stimulation. So jumping to our studies, or our study that we completed last year in, it was a pilot phase clinical trial of 2 subjects. So this here is spinal imaging of our first subject. You can see the artifact from his hardware, which is shown in MRI, shown as artifact, in CT shown as these bright rods and screws. And then in between the hardware, you can see where the spinal cord has been injured. And we placed the Medtronic array over the T1112L1 vertebrae, or within the T1112L1 vertebrae, in order to try to activate those same spinal segments that I was telling you about before, the L2 to L5/S1 regions. And after we implanted, we applied epidural stimulation during what we consider multimodal rehabilitation. So the subject would come in, we would set them up on a treadmill with bodyweight support and trainer assistance if needed. They would also do overground tasks with stimulation, such as standing activities or trying to step with a frame system or a walker. We would also have them do seated balance activities, so lean forward, lean back, flexion extension, as well as reaching and lateral movements. And then also we had them try to isolate single joint movements as well as whole leg synergistic movements in a horizontal, so minimal gravity influence, as well as in supine or prone positions, with stimulation on as well as stimulation off to look at the effects. So the rehab strategies that we employed while stimulating were to try to maximize the loading of the limb with minimal support, as well as maximize the speed while maintaining proper gait kinematics, with minimal trainer assistance provided that was still maintain safety, tried to minimize or prevent falls, that type of thing. And then it was a dynamic approach to each session with continuous adjustment, manual adjustment of stimulation parameters if we felt like the functional performance could be improved that we were observing. So jumping back to the evidence of dyscomplete SCI, over the course of training, so at session 24, 56, and 98, we shut the stimulation off, and in the sideline position, we asked the subjects to try to flex their left leg for a prolonged period of time, up to about 10 seconds, as well as try to flex just their right leg when that one was positioned on top of this board. At session 24, this subject only displayed the ability to activate his left leg muscles, but not his right. And this activity couldn't be sustained. It was more of a, I wouldn't say it was a spastic move. It was spastic, but it was very minimal movement, and it was maximal effort they were applying. So basically, we kind of described, try to make your best impression of Hulk. And at session 56, you can see the control of the left leg versus the right leg was improving, and by session 98, they could evoke one or the other with minimal movement. And over the course of 3 attempts, it was decreased to the point where the EMG activity was minimal as well. So it wasn't like a sustained and consistently repetitive activity, but we found it interesting that these spared networks that are associated with dyscomplete SCI were able to become a little bit more selective and more robust in their influence on the sublesional circuitry. So to jump to the functions that we saw enabled by epidural stimulation, in the early stages of our trial, we applied just single continuous programming of the stimulator while they were stepping on a treadmill with body weight support, trainer assistance at the hips, as well as at the knee and at the ankle. And the EMG patterns, as I showed from Manassian, you can see the rhythmic activity in the quadriceps. However, the medial hamstring showed very tonic or static activity during stimulation. By week 43, over the course of these weeks, we saw improvement in performance of stepping to the point where the subject did not need any body weight support, no trainer assistance at the legs, and they had their arms on these bars here for maintaining upright balance and weight shifting. The stimulation parameters that we began applying later on in the study involved trying to target the left side of the spinal cord as well as the right side of the spinal cord in an interleave pattern. And the reason we did this was because if we applied stimulation in this generalized symmetrical pattern, we saw spillover from one leg to the other where they could control the left leg, but the voltage or the frequency setting or the pulse width wasn't quite optimal for the other leg, so there'd be spillover in the effect. They'd try to flex one leg, both legs would flex. When we applied these parameters, we were able to adjust each side of the spinal cord, the stimulation that's being applied independently. And as you can see, the rhythmic activity in the quadriceps was still observed, but there was more of a rhythmic pattern in the medial hamstring in both legs, which we attributed to the ability to control these movements independent of trainer assistance. I can show a quick video here. So this is with no stimulation on. You can see the body weight support system, trainer at the hips, trainer at both knees and ankles, and the EMG. So these are EMG traces from the left leg and the right leg. Red traces are extensor, blue traces are flexors, and there's minimal activity. The small bursting you see may be caused just reflexively by the trainer moving their legs, proprioceptive input to the spinal cord. Now when we turn on the interleave stimulation, you can see the EMG activity is much more robust. There's rhythmic patterns that are alternating between flexor and extensor, and there's no trainer assistance at the hips, at the knees, no body weight support, and he's using these bars to try to shift his weight.
Hey, Peter, let's take just about a minute or two to wrap up, okay?
Okay. So one other point I was going to make was that with the dynamic rehabilitation that we were applying, we found that the subjects, when we applied an optimal standing performance configuration of stimulation, they could stand prolonged periods of time with just minimal effort to maintain their upright positioning. Then when we applied stimulation parameters that were involved with stepping, so slightly different frequency, slightly different electrode contacts, the subjects complained of knee instability, a lack of confidence in their ability to stand, and we saw rhythmic activity in the leg muscles. Then we applied those same parameters during stepping, and they could step independently. And then if we switch back to the other parameters, they could stand again confidently, independently. So this really emphasized that we saw a dynamic effect or effective training in a dynamic setting. So they could use suboptimal parameters to perform another function. However, applying the optimal parameters allowed optimal performance. So the networks are somewhat plastic and responsive to stimulation, as well as the rehab tests that are being applied. And I'll hop ahead and just kind of give a broad overview. Around the same time that we published this work on our first patient, both Gregor Cortine and Susie Harkema also published similar findings in additional cohorts of patients. So although our study was an N of 1 that we reported, in the broad spectrum of things, the results are very consistent. And so currently with COVID, our current trial in 2 participants to look at transcutaneous and epidural stimulation is on hold, but we hope to continue that study soon. And while in COVID hiatus, we're currently submitting a protocol to the IDE in order to study Asia A through D subjects with percutaneous temporary epidural as well as dorsal ganglion electrodes. And with that, I can take questions.
Excellent. Thanks, Peter. Really appreciate it. While we're queuing up Vivian, we're going to go ahead and there's a couple of questions, but then we'll have to leave some for the end. The first one was, could you just clarify the ASIA scores of the participants? I think you mentioned that right at the end.
Yeah, so
the Asian.
Sorry. So the first two subjects that we studied in the previous trial were ASIA A. So they had no function below their level of injury, either sensory or motor, and they were both thoracic level injuries. So I believe the first subject was T7 and the second subject was T3.
Okay, great. And then one other question I will, and then we'll get Vivian on up, was in regard to the duration of the stimulation for each session and how many total sessions per week and for how long were those sessions? So the duration of the sessions and how many times per week?
So the goal was to get in 3 sessions a week during a typical 5-day work week. And the rehab sessions were 2 to 3 hours long with stimulation applied only during the tasks. So like if we were switching between environments from treadmill to the mat, stimulation was not on during that time. The full session would last up to 3 hours. There were sessions where when we were recording data, the subject would be in the lab all day, but stimulation was not applied like the entire 8 hours. It was only during the task performance. Okay,
perfect. Well, thank you, Peter. A lot of these other questions, we will take those at the end. At this time, we're welcoming Vivian. We have your slides up, and I think we have your video going.
Yeah, can you hear me?
We can, yes.
Oh, wonderful.
And and technology actually is working for us, Vivian. So go ahead and take it away. Before you go ahead and start, sorry, there are some additional questions. We're going to try to get those at the end. But please, Vivian, please go ahead.
Yeah, thank you very much, Jen and Neski, for organizing this session and inviting me to participate. It's really wonderful to see everyone today. Now, in my talk, I will be building on the presentations from Chet and Peter and focus on the use of spinal stimulation for restoring functional walking after severe spinal cord injury. Now, as many of you probably know, a main research drive in my lab is to harness the networks in the spinal cord for restoring functional mobility after severe spinal cord injury. And for much of our work, we have focused on the lumbosacral enlargement, which is also the focus of this work to talk today. However, in other work, especially work related to persons with incomplete spinal cord injury, we have demonstrated that the cervical lumbar networks in the spinal cord also contribute greatly to the improvement of walking after spinal cord injury. So we have been activating these networks through simultaneous rhythmic arm and leg cycling, and more recently through the application of transcutaneous spinal stimulation. Now, this is going to be the topic for a future talk, but for today, I will focus on our work in harnessing the locomotor networks in the lumbar spinal cord to effectively restore functional walking. Now, you probably have heard me mention the term functional walking several times. The clinical definition of functional walking is coordinated and predictable movements of the legs that are weight-bearing during stance, that are propulsive to allow forward progression, and are fatigue-resistant to allow for reasonably long distances of walking that enable community ambulation. And we've focused our efforts over the years to restore walking after severe spinal cord injury, because at least 50% of persons with spinal cord injury never recover their ability to walk. And from our interactions with hundreds of stakeholders with spinal cord injury over the years, they consistently express to us a keen desire to walk again. So to understand how to activate the spinal cord to restore walking, we and others mapped the lumbosacral enlargement to understand where the locomotor related networks reside. And we and others found that these networks actually reside deep in the spinal cord, as you see on this cross section. So of course the question becomes, how do you activate them effectively? And a very wonderful approach that was already described by Peter is to use epidural stimulation applied to the dorsal surface of the spinal cord. And of course, several modeling studies have been done on this approach. And what you found out is that the stimulus from the epidural electrodes can penetrate about 200 microns into the dorsal surface of the spinal cord and likely activates the locomotor networks through indirect pathways. I'm not going to belabor what Peter showed, but he showed some very exciting results from his lab and others, where epidural stimulation in persons with severe spinal cord injury combined with physical therapy for about a year and a half, enabled people to step over ground for about 100 meters. And the reason why we're so excited about these findings is because they show that the locomotor networks are alive and well even after severe spinal cord injury. And our goal has been to improve access to the locomotor networks to meet the principles of functional overground walking. So our thought has been that activation of locomotor networks more directly through ultrafine penetrating electrodes may produce more functional walking. And we call this technique intraspinal microstimulation or deep brain stimulation. So to identify within these deep regions of the spinal cord where we really need to stimulate, we mapped the lumbosacral enlargement and identified the location of various components of the gait cycle in the ventral region of the spinal cord. These components are stereotypical synergies that make up the gait cycle for locomotion. And very interestingly, we found out that the functional map of these components of locomotor networks is not only repeatable from animal to animal, but they're also preserved between species. So you see very similar maps in rats, cats, pigs, and monkeys. And I'm showing just an example from a cat and a monkey in this slide. So now we know where to stimulate in this region of the spinal cord to activate various components of the locomotor cycle. So we developed a microdevice, the elements of which that you could see over here for implantation in the lumbosacral enlargement. A few microwires, each smaller than a human hair, are implanted in target locations guided by the map. An actual picture of the implant in an adult cat is shown over here. Now, this implant was tested chronically in rats and cats and acutely in pigs and monkeys. And what we found out is that these electrodes continue to produce the same responses again and again all throughout the period of implantation, suggesting that the implant is very stable. We also saw that the chronic implants cause minimal tissue reactivity. So it's a kind of a feasible and safe approach. And very interestingly, we found that through intraspinal microstimulation, you can produce reasonably long duration of stable and posturally appropriate walking. So we've got, or standing, we've got about 21 minutes of stable standing. Of course, the next question was, can we now use intraspinal microstimulation to produce functional walking? And for this, we chose the locations in the spinal cord that, when stimulated, produce upward, forward, downward, and backward synergies, which are the core elements of the gait cycle. And then we phasically cycled the stimulation through these microelectrodes to produce the gait cycle in both legs, and used feedback signals to modify the stimulation patterns as needed to produce a stable walking. And what you see over here is an example of the results produced by intraspinal microstimulation. This is an animal that is anesthetized and walking over an instrumented walkway. All movements in the highlights are produced by intraspinal microstimulation. And with only intraspinal microstimulation, these cats were able to walk across this 3.5 meter walkway 300 times, effectively equivalent to 1 kilometer of walking. And what's really exciting about this generated walking is that it does meet the requirements of functional walking. So there are coordinated movements of both legs. The walking is weight-bearing, propulsive, and fatigue-resistant. And the reason we're so excited about this is because 1 kilometer of walking is really unheard of using any neuromodulation device. I mean, this distance of walking is 10 times longer than anything out there. So really exciting. Let me wrap up just in the interest of time. We see that intraspinal microstimulation appears to tap into organized elements of the locomotor networks in the spinal cord. And the organization of these locomotor related networks is predictable and preserved between species. And with a very sparse implant, we can reliably produce complex functional movements, including weight-bearing, standing, and walking without an overt dependence on physical therapy. So what I think everyone would agree is that engaging the spinal networks is probably an effective means for improving mobility after spinal cord injury. For producing functional walking, properly precise localization of the electrodes and the pattern of activation may be very important. And what we're doing right now in collaboration with the Louisville group is trying to understand the sites and mechanisms of action of intraspinal microstimulation and epidural stimulation. And in a parallel study, we're preparing intraspinal microstimulation for first-in-human testing in persons with severe spinal cord injury. So exciting work taking place. I'd like to end by thanking the incredible number of people who've helped with this work and the generous support of multiple funding agencies. Thank you.
Thank you. Great presentation. There's definitely questions out there. So let me touch with the first one while we get John Chernesky up and queued up. One of the questions is, do you have any idea which neurons are activated by the ISMS?
Yes, in fact, very interestingly, Jan, from a biophysical perspective, intraspinal microstimulation and epidural stimulation act in the same way. In other words, they activate axons much more readily than cell bodies. So in the ventral region of the spinal cord, we found that intraspinal microstimulation activates fibers in passage within that network, within that region of the spinal cord. And these fibers of passage are really the basic structure for these networks that we are activating. So a single microelectrode can activate a whole network. So what we're activating is fibers in passage and interneurons that collectively synapse on motor neurons and activate them in a synergistic way and in a near normal order of fatigue-resistant activation.
Excellent. Thank you so much for that great talk. We're going to get to some of the other questions as we get to the end, but thank you. Our next up is John Chernesky. John is from the Praxis Institute, and he's going to talk a little bit about the spinal stimulation survey that we have going on. So John, we have you up on video. If you could share your screen so we can go ahead with your slides, that would be great.
Everybody seeing that?
We are good to go. Go ahead, John.
Awesome. Well, first of all, thank you, Jen, for inviting me to be a part of this fantastic workshop and presentation. And yeah, I'm going to give you guys a quick overview of understanding the end user and community and personal opinions and perspectives on spinal cord stimulation. What's going on here? There we go. So quickly in the next few minutes, objectives are to discuss the need for and lack of consumer perspective when it comes to spinal cord stimulation, to discuss the partnership methodology between the Praxis Spinal Cord Institute and the North American SCI Consortium, and then towards the end I'll share some preliminary results from the survey data, which is currently ongoing. So we took a quick look at the landscape of what's going on in spinal cord stimulation, and there's a lot of pieces that are moving. And we thought about what could the Praxis Spinal Cord Institute do to help contribute to this landscape and to provide an impact. And what we decided to focus on is the perspective of persons with lived experience of spinal cord injury, those that are living with spinal cord injuries, and what their thoughts are on the research that's being undertaken in this exciting area. You always practice what we call an integrated knowledge translation model, which is the meaningful engagement of the right research users at the right time throughout the research process. And this is a little postcard that we created for the ISCOS conference a couple years ago that discusses how you can engage people with lived experience throughout the entire research process to make sure that they're able to contribute a meaningful way to the work that's being done. So with that in mind, we reached out to the North American SCI Consortium, NASCIC, with a project partnership proposal, and they really were fantastic to work with and helped us go through the process of creating a good submission for the partnership. which included the level of engagement, the budget for the engagement of partners. And when it was approved, they recruited a great group of individuals with lived experience with spinal cord injury to assist us in this work. So what we ended up developing was the SCI Community Personal Opinions and Perspectives on Spinal Cord Stimulation Survey. There's a link to it right there if you're interested in completing it. It is only for individuals with lived experience and their direct care providers. Sorry, researchers, we love your perspective, but not for this particular survey. So the objectives were to identify the priorities for recovery, to gauge expectations for recovery, to better understand the willingness to participate in these therapies, gather opinions on clinical trial design, and assess demand on the availability of these treatment options and clinical studies. So the IKT model, what value did it actually add to the survey? A lot of different ways, but some of the ones we'll highlight here was in helping to develop the questions and better understand what actually mattered to people living with spinal cord injuries. Questions such as how soon after initial treatment would you be disappointed or regretful if you did not see any improvement? Separating the idea of a treatment versus what's currently being undertaken in clinical trial, and extending the survey to primary caregivers as well. You know, other options that were brought in, such as including the option of not wanting to change the current physical state of function. Again, a variety of different areas, but I won't go into too much detail given we're a little short on time. So far, what have we got? We've got about 220 odd people who have participated so far. About 65% of the respondents are male and 78% are a result of traumatic spinal cord injury. We've got about 70% in the US, 20% in Canada, and the remainder from different parts of the world. 81% have not had any experience whatsoever of spinal cord stimulation. 9% do have experience, and 11% have currently applied for a trial or have undertaken one. 38% of the respondents said that they have asked a healthcare professional about spinal cord stimulation, and only 1/4 said that the information they received was actually useful. This is, you know, the big stuff here. This is the priorities for recovery. As you can see, bowel and bladder function are far and away the most sought after areas of recovery, as well as standing and walking. Sexual function, followed by pain, fine motor function in the hands, spasticity management, balance, upper body function, including shoulder and arms, and blood flow. There were 17 potential priorities for improvement from therapy, and these are the top ones. But again, this is preliminary data. The survey is ongoing, and we'll certainly be looking to publish later when the survey closes out. Other questions were around participation in the clinical trial. The top 5 barriers to participation were the location, that they were unable to relocate to the study site for the duration of the study, the cost, lost wages, costs of relocating, etc., the distance to the study and the ability to, or the length of time that they'd be willing to travel, overall awareness of the therapy, knowing where these clinical trials were undertaken or what the potential risks or benefits might be, and personal support services required in order to undertake this therapy. Of those that responded, over half, 51%, were willing to relocate for up to 1 year. 97% were willing to follow a specific rehab training protocol. 50% wanted to do the training at a rehab facility. 40% actually thought that they would prefer to do it in a home setting. And in terms of the rehab protocol, most respondents were willing to undertake a 3 or 5 session per week, 2-hour per session training protocol. And of the interest in actually participating in a clinical trial, 83% responded that they would be willing to participate in a transcutaneous trial, and 67% were willing to participate in an epidural trial. So next steps, the survey will continue to be open until this Sunday, April 26th. So if you'd like to share it with your networks, we would very much appreciate it. You can find the link to the survey on our social media channels, Twitter or Facebook. or you can, maybe I'll share the link in the text box, chat box. We are developing a report for NASCIC, and for our website, we will do an open access scientific publication. All these will be done in partnership, including the data analysis and the publication with our partners at NASCIC, to actively disseminate the results to all SCI stakeholders, and the de-identified data will become publicly available if you want to parse through it. So with that, I'll just say thank you to our partners at NASCIC. The team included Ian Barkhart, Judith Smith, Shannon Sweeney, Rob Woodluck, and Jennifer French. There's the Praxis team, and additional thanks to Dr. Peter Grahn and Dr. Andrei Krasyakov, who assisted us in reviewing the survey. Thank you very much, and happy to take questions.
Excellent. Thanks. Thanks for your talk, John. That was really great. There are a couple questions about there, and I think you answered them right at the end of whether or not that data will be shared. Could you just reiterate for people where that will be, where we're planning on publishing it, and maybe what website they can go to once the data is published?
Yeah, to be honest, I'm not the lead on this project, so I'm not entirely sure, but I imagine that it'll be up on the NASCIC website. We'll share it with the team there. It'll also likely reside on the Praxis website, and depending on whether it's desired to be on an open data platform, we could also do that. So we're happy to share that information.
Excellent. Thank you. Thank you so much, John. Thanks for presenting that. We do have, before we get to additional questions, because there's definitely a lot of them out there, we just wanted to highlight Matthew Rodrick from Unite to Fight Paralysis. Matthew, I've unmuted you. You have just a quick, interesting tidbit that you can share with the audience of what you're doing in terms of your monthly calls regarding epidural stimulation, and then we can bring everybody back on while you're explaining that to answer some of the collective questions that there are. So Matthew?
Sure. Jen, can you hear me?
Yep, we can hear you.
Good. Well, as Jen introduced, thank you for the opportunity there. I'll start my video. Thanks for the opportunity to talk. For those who don't know me, my name is Matthew Rodrick. I'm the Executive Director of Unite to Fight Paralysis, National SCI grassroots advocacy organization. About 2.5 years ago, I'd engaged in several conversations with researchers and clinicians working on neuromodulation studies in human beings. And almost all of them told me they were often frustrated by the lack of opportunities for them to confer. And in the process of asking what that would look like, kind of became this idea hatched to facilitate, actually not monthly, as Jen mentioned at the front end of the meeting, but quarterly conference calls, quarterly web conferencing calls. We will have our 9th call this in a few weeks, and it's been extended. Early it was probably in the teens. Now we're up towards close to 30 who are invited to these conference calls. Initially, and for now, all investigators are those who are working on human studies. And the calls were initially really a forum to introduce and discuss what each of the participants were studying, what they were finding, questioning, concerned with. And after clearing a path, which wasn't altogether easy for the participants getting to know one another, the group landed on a few objectives. Amidst a wide range of subjects discussed, and in really no particular order, the primary drivers are in the conversation: how do we share work, preliminary discussions around an open data commons, also what is the strategy for translating discoveries to clinical relevance. Some have used the term low-hanging fruit, I'm sure you've heard. And then what's the nature of the work? More specifically, what are folks measuring? What can they agree on? What are the common metrics in order to coherently compare findings? And it's the last one where the group has really decided to place its current focus. In fact, our next call will be an attempt to whittle down a set of prioritized, coherent, and achievable metrics that the group can recommend to the rest of the field. Hopefully possibly in the form of a publication. And throughout, I think there's been a sense of frustration from many of the participants on the calls to make accurate, informative, comparative analyses from across labs, across groups, and how this may very well slow progress. The calls have thus far been, as I mentioned, with those who've been working in human beings, but you know, that could possibly change or be altered, or invite other folks to confer with this group as well. So hopefully that last point of focus for the last few calls will actually produce an output that will help move this field forward. And I guess the one last thing I'll say, and then I'll be done, is really just kind of a humble addition to the title of the talk. And it's really where we are today, where it's going, and to some degree how the SCI community can help in that progress. And I think this is an example, as John just articulated with NASCIC's efforts. This is sort of an effort in practice where those of us in the advocacy community can assist in facilitating progress, facilitating strategy, facilitating priority for this work. So thanks.
Excellent. Thank you, Matthew. And we posted in the chat box, if you're interested in joining his calls or learning more, we posted Matthew's email address, so you can get a hold of him directly regarding that. So with no further ado, with our short time left, we still have lots of questions out there. Before we get to them, I just wanted to mention everybody is please privately send me your email address. We'll curate those questions and make sure you get connected so that we can make sure that we answer all of the questions that are out there. Before we get to the serious questions, I do have to say that I think Peter won the background award for our Hollywood Squares event. There are a lot of questions out there about that, so congratulations, Peter. But let's get to some more kind of general questions that came up that I thought are really good for discussion. So one of them was regarding biomarkers. So for the people that are in these studies, are we testing their blood and trying to look for certain types of biomarkers that can determine success or failure?
It's a great question, whoever posted that. We are not yet at the University of Washington, but it's definitely something we'd like to do. Peter, how about you guys?
No, same. In our first study, we didn't collect any samples for analysis, such as markers of neuroplasticity. But moving forward, it's something that we plan to do. And
Vivian?
Yeah, we haven't done. I mean, I guess it depends on how you define biomarkers. If they are metabolomics, you know, if they're the omics round. We haven't done it in these studies. We've done it for different studies related to pressure ulcers. But for this one, all our biomarkers are either imaging or electrophysiological markers, and then we look at muscle and spinal cord histology.
Excellent, thank you. One other kind of general question that goes across all of the clinical trials that are going on is always a question of safety. So how are we looking in terms of safety of the participants, as well as are we finding that we're activating some other important structures that maybe you weren't anticipating during your trials?
Sure, I'll kick off there. We've had no serious adverse events with the transcutaneous stimulation. The one event related to the stimulation, we believe, was in one of our participants who resumed sweating, resumed diaphoresing below the injury. He also redeveloped a very mild skin rash, a little autoimmune rash, which he'd had, ironically, 12 years before his injury, and his family all has a history of this. And so while it was resolved with just topical anti-inflammatory cream, we view that as, again, evidence that the stimulation was restoring the autonomic system to stay closer to what it was prior to the injury.
Excellent. And for Vivian or Peter?
So for us, so for all the work that we've done with transcutaneous, be it spinal cord stimulation or therapeutic FES, we really haven't encountered anything over the many people that we've worked with. There was a single person who reacted to the electrodes, and we changed those to hypoallergenic electrodes. In terms of intraspinal stimulation, obviously that's still in animals, getting prepared hopefully for first in human. We have done extensive histology, try to understand the reactivity of the tissue to the implanted electrodes. And what is very important is how stiff the electrodes are. So if the electrodes are way too stiff, what happens is that the whole implant becomes a very stiff structure, and it's like a fork that's sitting in the spinal cord. So that very early on, when we used a stiffer microelectrodes, 50 micron in diameter and higher, that caused damage to spinal cord. But that would have been about 18 years ago when I moved to Alberta. Since then, we've moved to those much finer electrodes. We haven't seen any overt adverse event. Obviously, these are foreign bodies that are implanted in the spinal cord, and they do get encapsulated. But the encapsulation is actually very, very thin. So suggesting that the electrodes are well accepted by the tissue, and after the initial implantation and encapsulation, you don't end up with a continuous reactive, I guess, event. The tissue just encapsulates them and ignores them. So it seems to be safe using the parameters that we use, right, in terms of electrode diameters and so on and so forth.
And Peter, can you touch on safety and maybe some unexpected structures that may have been activated?
Yes, so with both the subjects that we've studied with epidural stimulation, as well as 2 subjects we've been working with transcutaneous, I'd say it's pretty consistent across all of them that in the early stages of stimulation, so the first few days when we're trialing things, they're much more sensitive to the stimulation. So like almost a sensation of discomfort when you're applying it, fluctuations in blood pressure, tightness, like shortness of breath, which we obviously immediately shut the stimulation off, let them recover, take vital signs, monitor them closely. But it seems to dissipate over the course of multiple sessions with stimulation and rehab, to the point where they either don't complain or they tolerate it, the stimulation, or in some cases, they can tell us like that stimulation isn't optimal, or this stimulation feels better, that type of sensory input, even though clinically they're complete injuries. They do describe some type of sensation when we're applying stim.
Excellent, thank you. And we're hitting on 5 o'clock, but I just want to end with a question for John. Looking at the preliminary results, what was an aha moment, and what were you desiring of how this can impact clinical trials going forward for people from the spinal cord injury community?
Mm-hmm. Yeah, I think as I mentioned, in the designing the questions, the idea of that people would not want a change in their current state. That surprised me, that people were quite satisfied, potentially satisfied with their current state of being. It wasn't something that crossed my mind. So again, adds to the value of integrated knowledge translation. So we're hoping that this survey will help to influence researchers' designs for their trials to better understand what the actual priorities for people with spinal cord injury actually are, and that will help to inform their work. In conversations with representatives from the FDA, they've clearly stated that in their approval processes, the risk-benefit analysis and input from people with lived experience is something they take into consideration. So hopefully that'll help to also influence the regulatory pathway.
Excellent. Well, we are out of time. And as we mentioned at the beginning, we're filling this with a lot of content in a very short period of time. So I'd like to thank our presenters, Chet, Peter, Vivian, John, and Matthew for joining us. And for everybody that came out today, thank you for taking the time on this topic. It's a hot topic. I'm sure it's going to be going on for more. And if you would like the slides, or if you would like your questions directed specifically to the speakers, please privately email me your email address. And that is at info@neurotechnetwork.org. Thank you so much, everyone. Please stay healthy and safe during this crazy time that we're in, but we're going to get it out together. So thank you. Have a great evening, morning, or afternoon, wherever you might be.
Thanks, everyone. Bye.